Okay, so now that we've talked about gross anatomy of the heart, we're going to talk about our next topic, which is heart physiology. And so, by way of reminder, if you recall when we did the tissue unit a few topics ago, we learned that there are four types of tissue in the body. So we had epithelial tissue, we have connective tissue, we have nervous tissue, and we have muscle tissue. And cardiac muscle tissue is going to fall under the realm of muscle tissue. So we've also talked about skeletal tissue or skeletal muscles. Smooth muscles are going to line the walls of our internal organs. And then cardiac is going to be the specific muscle tissue that's related to the heart. Some specific details about cardiac muscle. It is striated. So in that way, it's similar to skeletal muscle. In that it does have myofilaments present. And as a result, it's going to contract using the sliding filament mechanism, just like skeletal muscle does. Cardiac cells have 1 to 2 nuclei per cell, and we describe them as short, fat, branched, and they have this interconnected structure where plasma membranes of adjacent cells are going to form what kind of looks like corrugated cardboard connections. Okay, and that's what this would be right here. And we call those junctions intercalated discs. Okay, and this is specific to these junctions here between cardiac muscle cells. And this is what they look like. This is good images of cardiac muscle cells. You can see the striations that are present, the differences in the color changes. You can see how these cells are kind of thick and branching. You know, it's going to come out and it's going to branch into a couple different parts here. These darker areas here are going to be nuclei, and then the lines that you see. So here, this is an example of an intercalated disc between two cardiac muscle cells. So intercalated discs are going to have two types of specific structures associated with them. These are going to be two types of cell junctions, and the first one is going to be desmosomes. And if you recall, desmosomes are Velcro-like junctions that prevent cells from pulling apart during force. And so in the heart, this is important because the heart's going to contract, and that creates a lot of force or even shear on this tissue. And so desmosomes are important because they're able to resist shear, and the heart's able to contract strongly and not pull the muscle apart. The second type of cell junction that's present in intercalated discs are going to be gap junctions. And so this allows the cells, the cardiac myocytes, to communicate very quickly with one another. And so ions are able to pass directly from cell to cell through little corridors called connexons. And this allows ions to flow directly and quickly from cell to cell. And this enables the current, the action potential, to pass quickly through the entire heart so that the heart contracts all at once. Some other important features of cardiac muscle cells: they have lots and lots of mitochondria. So about 30% of the volume of the cell is mitochondria. This means that they're resistant to fatigue, but this also means that they have a increased need for oxygen. Okay, so they're generating ATP through aerobic means. They have a high need for oxygen, and they cannot live long without oxygen. They do contain myofibrils with sarcomeres and myofilaments, the actin-myosin. This gives it the striated appearance that we see in cardiac muscle cells. It does have T-tubules, and if you remember, these are invaginations of the sarcolemma or the plasma membrane of the cell, allowing the action potential to dive deep into the cell. And there is the presence of sarcoplasmic reticulum, which we know is a specific type of smooth endoplasmic reticulum that serves as a storage site for calcium. So here's all the features that we can see there. You can see the gap junctions that are going to help to connect the cells electrically so that ions can pass directly through. We have desmosomes there that are going to resist shear, and you can see they've got the sarcoplasmic reticulum for storage of calcium, as well as T-tubules for action potentials to be able to dive deep down into the cell. So here I've taken the time to compare skeletal muscle and cardiac muscle, and this is important. So I would recommend that as you're going through these to recall important facts about skeletal muscle to be able to compare skeletal to cardiac. So skeletal muscle is striated as well, so both skeletal and cardiac muscle fibers are striated and use sliding filament. Cardiac muscle is different, and we've talked about these, the structure and how these cells are arranged. We talked about intercalated discs containing or consisting of desmosomes and gap junctions and the functions for those. It is important to note too that these gap junctions, we talked about how it electrically connects the heart, and the term for that is called a functional syncytium. And this is the fact that all of the heart muscle is electrically connected through these gap junctions, which allow ions to flow quickly from one myocyte to the next, and it allows the heart to contract all at once. Okay, so just a broad overview of the general physiology of cardiac muscle. The heart has what's called an intrinsic cardiac conduction system, and this is the unique ability to be able to depolarize and contract without any external nerves. So if you think about that for a minute, this is very different from skeletal muscle. So skeletal muscle had somatic motor nerves to tell it to contract. So each skeletal muscle fiber had a nerve innervation telling it when to contract. This is very different. So heart muscle does have the ability to depolarize all on its own. It doesn't need a nerve to tell it to do that. There are two types of cardiac myocytes or cardiac muscle cells. These are pacemaker cells and contractile cells. And it's the pacemaker cells that are able to depolarize spontaneously. Okay, so basically they set the pace of the heart contraction, and that's why we call them pacemaker cells. There is a difference in cardiac pacemaker cells in that the influx of calcium causes depolarization. And if you recall, in skeletal muscle cells, it is the influx of sodium that causes depolarization. So this is a little bit different. So it's the influx of calcium. And we do have autonomic nerve innervation through sympathetic and parasympathetic input to the heart if the normal heart rhythm needs to be altered in some way. So it does have oversight, but it does have the ability to contract without nerves telling it to. So some myocytes are self-excitable, and these are the ones that we call pacemaker cells. Okay, so these are myocytes that spontaneously depolarize, and due to gap junctions between them, the pacemaker cells can not only initiate their own depolarization, but they can spread that depolarization through the rest of the heart. And this is what it looks like. So we would have kind of an area of pacemaker cells that are kind of located through here. And then as they depolarize, they're going to be able to spread that wave of depolarization throughout the rest of the heart muscle. Okay. This enables all of the cardiac muscle cells in the heart to be connected in what's called a functional syncytium. And this is the fact that the wave of depolarization travels throughout the entire heart, through gap junctions. Okay. This also means that either the whole heart contracts or it doesn't contract at all. Okay. So there's no small contractions versus big contractions depending on what you need. The heart always contracts as one unit. So there's no recruitment. It's not like skeletal muscle. Okay? If you remember, in skeletal muscle we had recruitment. So remember, if you were picking up a pencil versus, think if you were picking up a pencil versus a desk. Okay? So we were able to recruit based on the demand. The heart isn't like that. It always contracts as a unit. So it either contracts or it doesn't. Okay? In pacemaker cells, it's the slow influx of calcium through calcium channels in the plasma membrane. So it gets calcium from two different sources. It gets calcium from extracellular fluid, and it gets stored calcium from the sarcoplasmic reticulum. Okay, if you recall, skeletal muscle only got calcium from the sarcoplasmic reticulum. So only from sarcoplasmic reticulum. But in heart muscle, it gets calcium from two different sources. Okay? Another interesting fact about cardiac muscle cells is that cardiac muscle cells have very long absolute refractory periods. Okay, so this prevents tetany, or this ability to, generate a muscle contraction and then hold a sustained muscle contraction. We don't want that in the heart. Okay, so don't want tetany in the heart. Now, in skeletal muscle, we are able to have tetany because we have that happening of temporal summation. So we are able to temporally summate our skeletal muscle contractions because we can create larger and larger contractions. If you remember this type of diagram that we had, where each one of these was an additional stimulus that prompted that skeletal muscle to contract, and each contraction built on the one prior to that. And then eventually, if we stimulated, frequently enough, increase our frequency of stimulation, we are going to generate tetany. So skeletal muscle fibers have the ability to temporally summate. Cardiac muscle doesn't, because it's got a very long absolute refractory period. No matter what you do, it's not going to contract again unless it's given enough time. Okay, cardiac muscle cells, we talked about them before. They have a huge amount of mitochondria. So about 30% of the volume of the cell is mitochondria. This makes cardiac muscle cells highly resistant to fatigue as long as they have enough oxygen present. The bad part is that heart muscle is highly dependent on oxygen. So if you cut out the oxygen, so think, heart attack, where you decrease oxygen to that tissue is going to end up dying, and cardiac muscle cells do not regenerate. They're going to scar. So unlike skeletal muscle does have the ability to use anaerobic pathways to generate ATP. Cardiac muscle cells really don't, although they do burn um, other sources of fuel like lactic acid, but they do tend to be primarily aerobic respiration driven. Okay, so here's some differences between skeletal and cardiac muscle. There are two types of cardiac myocytes. We talked a little bit about the cardiac pacemaker cells. These are the ones that set the pace for heart contraction, and that's why we call them pacemaker cells. So this is actually very few of total cardiac cells. It's only about 1% of the total heart. And this is part of the intrinsic conduction system. And these cells are located in very specific locations in the heart. So this is known as the conduction pathway, and this is where these pacemaker cells are going to be located. The rest of the heart is going to be cardiac contractile cells. So these guys are responsible for the heart's contraction, or the pumping of the heart. And most of the heart is going to be this type of cell. Okay, so here's our pacemaker cells. Again, these only make up about 1% of the heart. These are the types of cells, these specific ones are able to depolarize spontaneously, and therefore they set the pace. What's unique about cardiac pacemaker cells is that they have an unstable resting potential. So they are constantly depolarizing towards their threshold. So if you compare that to skeletal muscle, here's skeletal muscle, it's sitting at its resting potential, let's say, of -70, right? And it's going to sit there until it's stimulated by a nerve. So here's our stimulus, and now it's going to depolarize and repolarize and come back to resting. It's got a very stable resting potential, and this would be skeletal. Okay, as you'll see, pacemaker cells aren't like that. They have an unstable resting potential, so that is different. And we have an action potential specific to pacemaker cells that consists of 3 steps. We have a pacemaker potential, which is a slow depolarization towards threshold. We have a depolarization, which is caused by influx of calcium, and then we're going to have a repolarization, which is caused by efflux of potassium. So here's step 1. Here's our pacemaker potential. And this is going to be this area here. If you look at this, this is our pacemaker potential. Okay, so this is what we mean by our unstable resting potential. It's constantly depolarizing towards threshold. Here's our threshold indicated here by this dotted line. Okay, so we have a slow depolarization towards threshold. And that's what we mean by the pacemaker potential, okay? And also, it's unstable. It's unstable resting potential. Okay, so it's not just a flat line like in skeletal muscle. This is caused by slow entry of sodium into the cell. So we have a slow depolarization because of sodium influx, okay, which is making the interior of the cell more positive. Step 2 is going to be our depolarization. So once we hit threshold, we're going to end up with our full-blown depolarization, which is going to be this part of the curve here. Okay, and this is due to influx of calcium. Okay, so we're going to have rapid influx of calcium. That's going to create depolarization in these cells. Okay. And then our third step is going to be repolarization. So at this point, what we're going to see is calcium channels are going to close and potassium channels are going to open. Okay, so we're going to have a repolarization. Potassium is going to efflux out of the cell, so we're going to be losing positively charged potassium ions, and that's going to end up restoring our negative membrane potential. Okay, so membrane potential is going to become more negative because we have potassium channels leaving the cell. Once the potassium channels close, then we have an opening of our slow sodium channels again. And this basically restarts our pacemaker potential. Okay, and then the whole thing's going to start over again. So this is what it looks like when we put it all together. So the important thing here is knowing that there's three different parts. Pacemaker potential is one, depolarization is a second step, and then repolarization is the third. Realizing too that that pacemaker potential, that slow depolarization is caused by sodium. Okay, our depolarization, after we reach threshold, we have our quick depolarization there being caused by calcium, and then our repolarization, which is being caused by potassium. So where are these cells located? So pacemaker cells, they're highly specialized. Remember, we talked about how they only account for about 1% of the total heart. Well, they're located in very specific areas, and we call this the intrinsic conduction pathway. And these are very specific areas throughout the heart. So the first area where these cardiac pacemaker cells are located is the sinoatrial node, otherwise known as the SA node. And this is going to be located here. It's in the right atrium. It's just below where the superior vena cava is going to dump into that right atrium. And cells, pacemaker cells in the SA node have the fastest depolarization rate. So this is actually what we call the pacemaker of the heart. It's going to set the pace because these cells depolarize the fastest. The next location is going to be the atrioventricular node, or AV node, and that's going to be located here. So this is basically along the inferior part of that interatrial septum. That was one of the landmarks that we talked about inside the heart. So right at the bottom of that interatrial septum, we have the AV node. The AV node and the SA node are connected by what's called the internodal pathway, and that would be this here. Okay, so here's our internodal pathway. So this is kind of a direct connection between the two, and then we're also going to have the ability to spread that wave of depolarization from the SA node throughout the atria. Now, once the impulse hits the AV node, it actually delays just a little bit. Okay, so we have this very small delay, and that's because we have the next structure where the pacemaker cells are located is called the atrioventricular bundle, or AV bundle, or bundle of His. Okay, and this is located right in this area through here in the top part of the interventricular septum, and that was another one of the landmarks that we talked about before. This is the wall in between the two ventricles. So at the top of the interventricular septum is the AV bundle, and this is very special because it is the only connection between the atria and the ventricles. So we know that we have gap junctions connecting cells, and it allows for electrical connections between cells. But those cells are all connected. The atria are all connected through gap junctions, and the ventricles are all connected through gap junctions, but actually there's no connection between the atria and the ventricles. Okay, there's not a lot of gap junctions through here. Okay, the only connection between the atria and the ventricles, we've kind of got one line here, and that's going to be the bundle of His, or the AV bundle. And so basically it's like we're taking this major highway and we're bringing it down to like this one little country road that's providing a connection between the atria and the ventricles. So it ends up slowing down the impulse. We have a slight delay and then a slowing of the impulse, and then we get down to the ventricles, and it can spread through gap junctions throughout the ventricles. So also too, think about it. What would happen, let's say that you had a stroke, or a heart attack, and you knocked out the AV bundle. Imagine how debilitating that would be, because now you've lost that connection between atria and ventricles, right? It's just down to that one little area there. So it's kind of interesting. And so now what's going to happen is that the bundle of His is going to branch. So you can see it's going to come down, and now in that interventricular septum, it's going to branch out into a left and a right bundle branch. Okay, and that's what we see here. Left and right bundle branches. These are going to take that impulse down the interventricular septum. And then when we get to the bottom or the apex of the heart here, they're going to turn and travel out through the walls of the ventricle. And now that impulse is going to be conveyed throughout the lateral walls of the ventricle, and the walls of the ventricle are going to contract. So we call these Purkinje fibers, otherwise known as our subendocardial conducting network. And these fibers are going to create contraction throughout the walls of the ventricles. You're going to see these fibers come up through the papillary muscles. Remember those papillary muscles where the chordae tendineae come down and attach? And what we see with this is that we have much more diffuse or kind of a much more pervasive Purkinje fiber arrangement on the left side than the right side. So the right side needs a little help. So that's where the moderator band comes in. So moderator band has some of these Purkinje fibers traveling through it. And if you remember, moderator band is going to connect the interventricular septum to the right lateral ventricle wall. Okay, so it's basically kind of like this direct connection heading over. So we've got Purkinje fibers that are going to conduct that impulse directly over to the wall too. It just needs a little help on the right side. Okay, so all of these areas that we just talked about are areas where the pacemaker cells are located. And when we talk about them all together, they create the cardiac conduction system. So cells in this area are all pacemaker cells, and they are able to generate contractions. So we can call this flow of depolarization our sequence of excitation. And this is basically how the impulses move from one location to the next. So if you go through this list, these are all the locations we just talked about. These are locations of pacemaker cells. SA node, which is responsible for generating the sinus rhythm, or kind of our base rhythm that our heart contracts. Our internodal pathway, that's going to connect SA node to AV node. Our AV node, we're going to have a slight pause there. The impulse is going to travel through the AV bundle. We've got left and right bundle branches, and then we've got Purkinje fibers conducting that impulse around the walls of the lateral ventricles. The amount of time it takes for that impulse to travel from SA node all the way down to the last ventricular muscle fiber is 0.22 seconds. Right. And this walks you through it again as well. So this is kind of our sequence of excitation. It's going to travel through this. Every time the heart beats, this is happening. Mhm. And just by reminder, the atria and ventricles do not have gap junctions between them. The only electrical connection they have is through this little guy right here, this AV bundle. Okay, and so that's going to be the only electrical connection between atria and ventricles. What happens if your cardiac pacemaker cells don't work? So what if you have a heart attack or something happens and they don't contract like they're supposed to? Well, this would be a case where you would have patients with pacemakers. And so they're implanted, they're about the size of a quarter, and they're going to put them right underneath the skin, and then they're going to feed the wires down into the heart, typically into the right side of the heart, and it's going to provide some external stimulation. Sometimes you have this, they're called demand pacemakers. And so they're only going to, start initiating a contraction when you need it. So they don't necessarily stimulate contraction all the time, but if your heart misses a beat, it's going to then take over and stimulate a heartbeat.